up guys, BIOBEAN here. Today we're going to be talking about DNA damage, so let's get started. DNA damage or changes to the DNA can occur from a variety of factors, and we break those factors up into two groups, being exogenous factors and endogenous factors. So looking at the beginning of the word exo and endo, we can infer what those different factors mean. So exogenous factors, it has that root beginning word exo. So that means factors are coming from the external environment. So there are a variety of different exogenous factors that may cause DNA damage. So that first one is UV light. So that's some of the light that comes from the sun is UV light, and that is why we need to protect our skin using either clothing or sunscreen, because that UV light coming from the sun is capable of causing DNA damage that may then lead to cancer. The second exogenous factor that may lead to DNA damage is chemicals. So there are a variety of chemicals that have the property of causing DNA damage. We generally know those as carcinogenic chemicals. So not all chemicals are capable of causing DNA damage. It is only particular chemicals that have that property. And the third exogenous factor that may lead to DNA damage is ionizing radiation. So that's radiation in the light spectrum anywhere from UV onwards. So that includes X-rays and gamma rays as well. So anywhere in that spectrum of the light, it may cause DNA damage. And that's why generally when you visit a dentist and you get an X-ray, they give you that giant lead blanket thing, and that is to protect the rest of your body from potentially absorbing that ionizing radiation and inducing DNA damage. All right, so now on to the endogenous factors that may lead to DNA damage. So if exo means external or outside factors, then endo means internal factors. So these are the internal factors that drive DNA damage within our bodies. The first endogenous factor that may lead to DNA damage is reactive oxygen species that is generated in the mitochondria during cellular respiration.
Okay guys, so I messed up here a bit. So for Ross, a normal oxygen exists in our body as O2. However, some processes, such as during cellular respiration in the mitochondria, that O2 can pick up an additional electron that causes it to now be reactive.
This is now a very reactive oxygen because it'll now go and release its electron to other molecules and create a chain reaction. So these reactive oxygen species, or ROS, can go and then affect the DNA and oxidize the DNA in a way that causes DNA damage. The second endogenous factor that may lead to DNA damage is errors in replication of the DNA. So as the polymerase is going through and trying to replicate the DNA, there can be errors introduced that will cause DNA damage. So we're going to focus in on the endogenous factor of errors during replication and how that may be a source of DNA damage. So how are errors introduced during DNA replication? So here we have a little piece of DNA, and it's going the 5' to 3' direction, and so our polymerase is going to be running along this piece of DNA, synthesizing the new strand along it. And so the polymerase, as it's sliding along, is reading each nucleotide on the template strand and putting that complementary base, following the base pairing rules, on that new strand. So what happens if the polymerase adds the wrong base? So looking at our template and newly synthesized strand, we can see that DNA polymerase put the wrong base down. So here there is a G, and that means that should have been a C on the new strand. However, polymerase made a mistake and it put a thymine. So now, because they're not properly base paired, the hydrogen bonding is not matching up, it's going to cause an actual bend in the phosphate backbone. So it's actually going to be a noticeable bump within the DNA chain because the G and the T are not properly base pairing and they're causing that bulge. Now, generally, polymerase is able to detect this issue because it has proofreading ability. So the back portion of the polymerase, as it's adding the new bases on, will be able to detect the mismatch and actually use its 3' to 5' exonuclease ability. So again, breaking up that word, exonuclease. So the exo part means from the outside, and the nuclease means that it removes the nucleotides. So it has a 3 to 5 prime exonuclease capability. So what that means is as DNA polymerase 3 is running along this chain, it will notice that it put the wrong base on, and then it will slowly chew up the bases from the 3' end until it meets that mismatch. So DNA polymerase would quite literally go back and use its 3 to 5' exonuclease ability, and it will remove all of the incorrect bases until it finds the mismatch. Now, once it's found the mismatch, it will go on and continue to add bases in the correct way. So our polymerase has gone, chewed up all of those wrong bases, and now it has laid down the correct base where the mismatch was, and now there is no longer any bump within that DNA backbone. But what happens if polymerase misses that mismatch? So if the polymerase does not detect the mismatch as it's doing the replication and it continues to move along, then there is a particular mechanism to correct this mismatch, and this is called mismatch repair. So this employs endonucleases to cut within the middle of the DNA strand to remove the base that is not correctly paired with the template strand. And so when we look at the word endonuclease, we can break it up into the first part and the second part of the word. So the first part of the word is endo, so that means from inside. And then the second part is nuclease, and so that means it removes the nucleotides off. So the endonuclease works within the inside of the DNA strand, and it can make 2 single-stranded nicks within the backbone to remove the mismatch base. So it's quite literally going to make a nick at either side of the mismatch, and it is going to remove that base that is incorrectly paired. Then there is going to be a polymerase that comes and fills in this gap and applies the correct base. Now that the mismatch repair endonucleases came and excised that mismatched base, and a polymerase came and laid down the correct base, there is now no longer a bulge within the DNA backbone, and it is now a correct strand of DNA, and there is no longer any DNA damage present. So something I want to remind you is that mismatch is not a mutation. So a mutation is a double-stranded change in the DNA. But when we have a mismatch, it's only a single-stranded change. So it is considered DNA damage, but not a mutation. And so the only way for DNA damage of this kind to lead to a mutation is if it is left uncorrected for multiple rounds of replication. So if this error was allowed to persist after multiple rounds of replication, we would first see this set of DNA damage within the first daughter cell that was created. So this would be created in the parent cell during S phase of the cell cycle. It would then go through mitosis and get split off into one of the 2 daughter cells that are created at the end of mitosis. So this will only become DNA damage in one of the daughter cells following 2 rounds of replication. So after that first round of replication, it inherited that mismatch, but it was only a mismatch on that one strand. So now we're doing replication in that daughter cell that was created. So in this daughter cell, those 2 strands are going to be pulled apart. And now the one strand that was the original strand that was being copied in that original cell, it's still going to have that correct DNA sequence. It was always supposed to have a G there. And so when I go to replicate this strand, it's going to create the correct strand that should have been there originally. So here now, after that second round of replication in that daughter cell, this strand is going to be correct, and there is going to be no DNA damage and no mismatch because it was following that original correct template strand. And so this will be inherited into a daughter cell, and it will be completely normal without any mismatch or any DNA damage or mutation. But now, this strand that was created in that original cell that had the mismatch is going to cause a DNA mutation. So now look at the new strand that was created off of this, what is now a template strand, but what used to be the newly synthesized strand in the daughter cell. It now has a double-stranded change. So here, where it should have had a G and a C, it now has an A and a T. Because this mismatch where the T was being paired with the G was never properly corrected, now when this strand is pulled apart and used as the template strand, DNA polymerase will never know that there was that mismatch there because it just sees that template strand by itself. So it will never detect that bulge in the DNA. And so now it is going to see that there is a T down here. It doesn't know that that T should really be a C, and so therefore it's going to put an A on top of it as the complementary. So now when the cells go to do cytokinesis, these are going to be separated into two separate cells. So now after 2 rounds of replication, we're left with a daughter cell that is completely normal and a daughter cell that has a mutation where there was a single base change on both strands. So because it is on both strands, it is a mutation. And so a mismatch, if allowed to continue across replications after 2 rounds of replication, it will give a single daughter cell that has a mutation. Another type of DNA damage is thymine dimers. So again, it's called DNA damage because it's only going to be damage associated with a single strand of DNA. So if we had damage on both strands of DNA, that would be called a mutation. So when 2 thymines are together on the same strand and they're exposed to UV light, they may covalently bond with each other. So when these 2 thymines pair together on the same strand are exposed to UV light, it can actually break the hydrogen bonding with their base pair on the complementary strand, and it can cause them to covalently bond with each other. So they're actually going to bond with each other. And this is called a thymine dimer, and this is bad because it no longer is base paired with its complementary base, and it is a source of DNA damage. These thymine dimers can also be very damaging because they will hold up the polymerase during replication. So polymerase will run up to these thymine dimers and will not be able to push through them because they are covalently bonded to each other. So it's something that must be repaired during G1 of the cell cycle before replication can happen. So that is one of the things that is being done during G1 of the cell cycle, is making sure that the DNA is all ready for replication. So one of those things is fixing these thymine dimers. Some organisms are able to repair this using the protein photolyase in combination with white light. Organisms that possess this photolyase, when exposed to white light, they're able to break the covalent bond between the 2 thymine dimers, and that reforms the hydrogen bonds with its base pair. And this then repairs the thymine dimers so that replication can continue in S phase of the cell cycle. But humans do not possess photolyase and therefore have had to come up with a different way to repair these thymine dimers. We use what's called excision repair. So we have endonucleases. So if you remember, endonucleases are nucleases that are able to cut within the middle of the DNA strand, and they will actually make a nick on either side of the thymine dimer and remove that thymines and then lay down new thymines that are not covalently bonded with each other. So we just talked about DNA damage, which is DNA damage that occurs on only a single strand of DNA. But what happens when we have double-stranded breaks? So that's when you get a break in the DNA backbone on both strands. So this usually occurs when you're exposed to UV light. So UV light can cause double-stranded breaks within your DNA. So these double-stranded breaks are repaired during G1 of the cell cycle. So again, this is the stage where the cells are making sure that their DNA is able to be replicated. So if there is a double-stranded break, it would replicate them as if they're 2 independent chromosomes, and you would actually increase your chromosome number if you allowed replication to continue with that double-stranded break. So that needs to be repaired before we actually do replication. So this is repaired through multiple mechanisms, but we're going to be focusing on non-homologous end joining. So these double-stranded breaks are repaired through non-homologous end joining. So what that means is that there are proteins that detect that the double-stranded break has occurred. They bind to the ends of the DNA where the break has been detected, and they quite literally ram into each other and try and connect the 2 strands back together. So they're not worried, maybe when the double-stranded break happened, maybe we lost one of the bases. Maybe during the slamming together, there are some bases that kind of fall off because it's quite literally slammed together. And the DNA, the mechanism doesn't really care about that. So non-homologous end joining tends to lead to mutations because they don't really care about if there was information loss and trying to regain that information or trying to repair any damage that may have occurred. It's just trying to combine those 2 strands back together to create one linear chromosome again. So this is more error prone and may lead to a bit of lost DNA, but it solves the issue of needing to combine these two strands together. So that concludes our video on DNA damage, where we talked about two main sources of DNA damage, as well as a source of mutation being double-stranded breaks and how those are repaired. If you have any questions about anything that we went over or anything related to biology, please visit biologybean.com, where you can access all of your tutoring needs, tutored by yours truly. Please don't forget to comment, like, and subscribe. It would be really appreciated.